the second lecture in our biochemistry module, we're going to talk about enzymes, possibly the most important proteins in biological systems. We'll start first with fundamentals. Enzymes are noted as biological catalysts. They are unchanged by reactions, excuse the typo, and are reusable. They also do not alter enthalpy or Gibbs, but they do alter the reaction rate. Catalysts do not alter enthalpy and Gibbs, and it's all about the rates. They're also highly specific and they're exergonic, meaning they decrease your energy needed for spontaneity. Enzyme shape is also directly related to their function. This is true for all proteins. Protein function is always linked towards their structure. The temperature, salinity, and the pH can denature these enzymes, which also results in a loss of function. Take note that this is true for all proteins, though. The types of enzymes that you'll commonly see on the MCAT are listed below. The name of the enzyme can often give away what their function is. Oxidoreductase, as the name suggests, will undergo some sort of oxidation-reduction reaction on its substrate. Long story short, there's going to be some form of electron transfer occurring at the active site. Transferases will transfer or move functional groups. Hydrolases add water, thus cutting substrates. Lyases cut without water. Isomerase is the interconversion of isomers and stereoisomers, and ligases join two larger molecules together. Oftentimes, the names of these enzymes are going to say what the function is, so just take note of that. You don't have to memorize every single enzyme that you see, because there's clearly too many, but having a general idea of what each name means is going to help you a lot. The enzyme mechanism is simply done by three different types. There's a stabilization of transition states, allowing for a favorable microenvironment, or bonding with the substrate temporarily. Certain enzymes do require cofactors or coenzymes. This can be done at the active site or at allosteric sites, which we'll get into later on. The active site is noted as where the enzyme function is determined. This is where the catalysis is facilitated. If I were to have an enzyme and it were to set in with a given active site, it would be specific towards one substrate, therefore allowing for a specific function. The active site is what determines the function of the enzyme, so whenever you think about what enzymes do, the active site should be the first thing that comes into mind. So in this case, for this active site, there's only one substrate that'll fit, therefore making this specific. When we talk about the active site, there's two different theories on how the mechanism works, and there really is no agreement as to which one it is. The lock and key mechanism states that enzyme substrate will fit with exact complementarity. So as I drew in the previous lecture, the shape of the substrate fits exactly in the active site, thus allowing for function. The induced fit mechanism, on the other hand, says that the enzyme substrate bonding will change the enzyme conformation temporarily. We can't really agree on which is which and what the true active site mechanism is. Certain enzymes exhibit more of one character than the other, but in general, it's a little bit of both. We can't really talk about enzymes without talking about enzyme kinetics, so we'll go into that in the next portion. There's several mathematical representations that can be used to talk about enzyme kinetics, and we have to graph them accordingly in order to have a good representation of what it is. The first and arguably most important equation is the Michaelis-Menten equation, which is given below. The Michaelis-Menten equation, quite simply, is showing the velocity of the reaction. A quantity that's important when we're talking about the velocity of the reaction is Km, which is simply stated as the substrate concentration at which half of the enzyme active site is full. This is also where we would say that the velocity max is at half. Oftentimes, when you plot the Michaelis-Menten relationship, it's going to be substrate concentration versus reaction velocity. As you saw from the Michaelis-Menten equation, it's not exactly nice to look at times, so there's other ways to represent the graph to show a little bit easier data to analyze. The Lineweaver-Burke plot is the double reciprocal graph of a Michaelis-Menten equation. Now, just by looking at the graph, why might this be advantageous over the Michaelis-Menten equation? Well, it's linear for starters, which makes it a little bit easier to analyze the data. Also, the MCAT is going to ask for certain quantities, such as what the slope of this equation is. You can figure this out just by manipulating the Michaelis-Menten equation, though. One more representation of enzyme kinetics is cooperativity. This is sigmoidal because of these cooperations between multiple active sites. Again, the most common that you'll see on the MCAT is hemoglobin. And if you remember what hemoglobin looks like, it's got multiple heme groups in complex with irons in the center. If one of these binds, then it's going to create a cascade reaction where the other ones are going to have a higher affinity, therefore having an exponential increase, which is exhibited below. When it comes to calculating these sorts of relationships, you're not going to have to do that directly on the MCAT, but understanding what cooperativity entails is going to be important. The last bit of our lecture is going to talk about enzyme regulations and what it entails. The most common way that enzymes are regulated is by feedback inhibition, which is simply a regulation by concentration of products in the pathway. Below is a diagram of this. When you look at a pathway, there are multiple enzymes and intermediates along the way. What you'll notice though is as you have an increase in products, it's going to inhibit enzymes further up on the chain, thus regulating concentration. The opposite is true. If there is a decrease in products, then you're going to see the opposite effect occurring, and the pathway is going to proceed more aggressively. When it comes to most biological systems, there is going to be some sort of feedback inhibition in place. The other types of inhibitions you'll see are called reversible inhibitions because they can be obviously reversed or counteracted. There's a lot of these, and they're going to change the graphical representations of the graph. I have listed the changes below and what these entail, but I have a really good metaphor for remembering these. When you think about reversible inhibitions, you should think about speed dating. If you've ever gone or seen a speed dating area, what you have are a bunch of chairs with people sitting at them and other people cycling through via stations. What you can think of is that the people sitting at these tables are the enzymes, and the people sitting are the substrates. So you have a fixed amount of people walking around and a fixed amount of people that will pair with these people walking around at tables to, well, speed date. Think about the active site as the chair in front of these people that are sitting down. With that little picture set up, let's go into each type and how we can use the metaphor to represent these. Competitive inhibition is when the inhibitor binds to the active site. Because of this, the Vmax is unchanged and the Km increases. Now note that the Km can therefore be understood as the substrate concentration at which half of the enzyme's active sites are full. So let's look at it this way. If we've got a certain number of tables and chairs and a certain number of people walking around, everything's good. But what if I were to introduce more unruly individuals that were taking up these chairs? So if that were the case, what would happen is the likelihood of these chairs being filled would increase. However, the speed at which people date is going to be the same. Therefore, the Km is going to increase, but the Vmax, the speed at which you see other people during this little dating expedition, is not going to change. This is what we call competitive inhibition because the inhibitor is going to go directly to the site. So in the metaphorical example, if you add additional people, then you're going to have a less likelihood of a given substrate sitting at those chairs. A non-competitive inhibition is when the inhibitor binds with equal affinity to the enzyme and the enzyme substrate complex. So this is going to decrease the Vmax, but it's not going to change the Km because of the binding to equal affinity. Think about this as a person that just sort of third wheels and butts in when someone's sitting down or not. They really are indiscriminate about this. So whether there's a person sitting in that chair and actively having a conversation with the person sitting or not is not going to affect whether or not it's going to go in. Because of this, the speed at which these enzymes or the people sitting is going to decrease because they're not going to be able to go through as many people with these distractions going around. The Km, however, is unchanged because, like I said earlier, this person is not going to really discriminate as to whether the chair is full or not. A mixed inhibition is when the inhibitor binds with unequal affinity to the enzyme and enzyme substrate complex. This is also going to decrease the Vmax, and the Km is going to change depending on the inhibitor. Think of a mixed inhibitor as an inhibitor that is going to be partial towards the enzyme or the enzyme substrate, but can still do both. So again, a person that will butt in and third wheel whether or not a person's there, or will go in on their own, but it's going to have preference into one state or the other. An uncompetitive inhibition is when the inhibitor binds only to the enzyme substrate complex. So think of this as a person that doesn't have the confidence to sit down and approach somebody directly, but will gladly butt in if someone else is talking. So this is going to decrease the Vmax and the Km because as he's butting in and interrupting, the speed at which these people can go through and the speed at which that person sitting down can see individuals in the speed dating is going to decrease. Also, the Km is going to decrease because the other people are going to notice that this is happening and thus be less likely to sit down. Irreversible inhibition is when the inhibitor prevents the enzyme from functioning permanently or for a long duration. I tend to like to think of irreversible inhibition as if somebody spills wine on, spills wine on your dress, or if you get injured and you say screw it and you decide to go home. Therefore, you're not dating. Therefore, you're not functioning. Note that irreversible inhibition, although the name says it's irreversible, there are ways to go back to a functional state. However, it's much more difficult than the other types of reversible inhibitions. Lastly, let's go into some regulatory enzymes. There are different ways that enzymes are regulated, and there's three in particular that you'll see on the MCAT. Allosteric enzymes have sites that can be occupied by activators to increase or decrease affinity. So if you remember, if I were to have an enzyme like so, the active site is listed here, but there could be an allosteric site somewhere else in the enzyme. Depending on whether or not there is some sort of activator or deactivator, some sort of cofactor is going to attach here, and that's going to affect the active site indirectly. That's what it means by allosteric regulation. Covalent modification is oftentimes glycosylation, phosphorylation, or dephosphorylation. So glycosylation, as the name would suggest, is the addition of carbs. Phosphorylation and dephosphorylation have to do with phosphate groups. So enzymes are going to want to pick up these sorts of modifications somewhere along the way to enhance or decrease their function. Zymogens, lastly, are enzymes that are secreted in an inactive form. They're activated by some sort of cleavage. Now, you're probably wondering, why would a biological system elect to do this? Well, if we can produce a larger molecule and just split it later, we could get additional enzyme or additional protein without taking up as much room as it would to make two of these. It also operates less cellular machinery, so it's more efficient this way. Okay, so when it comes to the biology section on the MCAT, you're going to see a lot of enzyme kinetics and enzyme regulation and inhibition coming up. So this lecture in particular and this topic in particular are going to be incredibly important come test day. In general, biochemistry is a very big field in medicine, so the MCAT is going to reflect this, so expect to see a lot of that. In our third lecture, we're going to go into non-enzymatic protein function and protein analysis, so we'll see you then.